Hi, Sarah.
Oh,
Very good to see you.
nice to see you too.
Thank you very much for joining us today. So we're really looking forward to hearing your talk about your recent paper in molecular biology of the cell on B-cell receptor signaling. Thank you very much.
Well, thanks for having me.
Also, can you maybe introduce yourself with one or two sentences?
One or two sentences. Yeah, my name is Sarah Veatch. I'm an associate professor in the biophysics program at the University of Michigan. I'm going to introduce a little bit about what our lab does, but generally interested in how kind of phase separation in the membrane and domains can influence kind of signaling pathways in cells. So it's wonderful to have a chance to talk about this paper that came out earlier this year. This was the thesis work of Marcos Nunes, who graduated about a year ago around now and was able to participate in graduation ceremonies, which have been unfortunately canceled this year. But anyways, so as I said, my lab is interested in kind of understanding if and how kind of this phase transition that happens in membranes that causes these liquids to demix, how this could contribute to cellular functions. If it's there and how does it, if it's there, how does it do this? Our kind of main approach in the lab is to use fluorescence localization imaging, often referred to as palm and storm, to kind of test hypotheses that come about from kind of this physical understanding of phase separation, but in the intact. So one of the projects that we focus on in the lab involves why do cells, immune cells, in particular B cells, activate when their B cell receptor or their immune receptors are clustered. And so here's an example of a B cell that's just being imaged by microscopy, and we add a cross-linking ligand at time equals 0, and you see these dramatic clusters form. Not only do these receptors cluster, but also all sorts of other cellular events happen. Things like calcium levels rise in the cytoplasm. Lots of proteins change their activities and get phosphorylated and get modified. And then there's downstream events that kind of impact things like cell cycle and cell differentiation and antibody production. Now, it's kind of interesting that you can cluster these B cell receptors either by a real ligand, so something it's naturally kind of been developed against, or just an antibody or streptavidin or something that's really not very biological. These clustering agents can be on a surface or in solution. There's all sorts of different ways it can be presented to the cells and they still respond. And at the end of the day, what has to happen for these cells to respond is that these receptors have to get tyrosine phosphorylated. So something about clustering these receptors leads to their being phosphorylated. Now, in a very overly simplistic model of this, you think about the membrane of these cells, not just containing receptors, but they also contain regulatory proteins. Many of them, but the two that I'm going to think about today are a kinase called Lin and a phosphatase called CD45. And in a kind of a normal state of these cells where everything is floating around, the kinase is responsible for phosphorylating the receptor, and the phosphatase dephosphorylates the receptor. And these things are all working all the time, fighting against each other. And at the end of the day, the phosphatase wins. Okay, so that on average, the receptor is not going to be phosphorylated. But for some reason, when you cluster the B cell receptor, this tips the scale so that the kinase can win. So then the receptor ends up getting phosphorylated. And this phosphorylation of this receptor is kind of sensed by other proteins in the cell that kind of facilitate this kind of downstream cellular-wide response. So one of kind of why this happens is one of the big open questions still, I think, in immunology, and not in just for B cells, but for other types of cells. And kind of one class of ideas that's been thrown out there is that for some reason, clustering the receptor shifts the local concentration of these kinases and phosphatases. And this has been thought about in a lot of systems. And here's an example in a T cell receptor system. But basically, in that case, the kinase has a different name, but has the same properties. And the idea is that you can go from low phosphorylation to high phosphorylation of receptor by either increasing the kinase concentration or decreasing the phosphatase concentration, or some combination of it. So that just because this is a biochemical reaction is going to tip the scale so that on average you're going to get more phosphorylated receptor. Okay, so how could you shift this local concentration? Again, there's many ideas that are out there already, and one of them is that by clustering the receptor, it partitions into kind of a more ordered membrane domain, commonly referred to as a lipid raft, and that these raft domains kind of enrich in this kinase, and they protect the receptor from the phosphatase, and that facilitates the kinase winning. Another idea that is also quite popular is that maybe there's something about the clustering of the receptors or the engaging of the receptors that occludes the phosphatase, right? So maybe there's some reason why the phosphatase can no longer fit into these clusters, and that will lower the phosphatase concentration and also promote the kinase winning. And one way that kind of this is, you could definitely easily imagine this happening is if you're being stimulated by a surface, and so the receptor is interacting with a surface, it's being held in close proximity to a surface, and these phosphatases often have really bulky extracellular domains, and maybe they're just so bulky that they can't fit into this space anymore. So this idea is commonly referred to as this kinetic segregation model. But both of these ideas have been around for a very long time in this field. Okay, so our methods are super resolution imaging. Just a quick primer of what this looks like is we'll label a receptor or a protein or a peptide with a fluorescent marker on a cell. And if we look at all of the markers at all of these fluorophores at the same time, you get a pretty picture, but it's a little bit blurry. If you can find a way to turn most of those fluorophores off and just look at a few at a time, you get an image that's basically a lot of spots, which itself isn't very informative. But you can localize each of those spots to much higher precision than its width. So you know where the fluorescence is coming from, even though it's a diffraction limited image. Seeing a few spots isn't very helpful, but if there's a trick to kind of cycle through and look at all of the spots, then you can spatially sample the whole cell. We do lots of image processing to sort all this out. But at the end of the day, you have kind of a list of localized positions of all these molecules, and you can use that to reconstruct what's called a super resolved image. And so we get about an order of magnitude increase in lateral resolution with this trick. All of the data that I'm going to show you today, I think, involves looking at two colors and not just one. And so it's just the same thing. We just do the same thing with two probes at the same time, basically, and we look at them on different channels, different parts of our camera. And then we end up with kind of these pictures that are just a whole bunch of spots. And we quantify these spots with something called the pair cross correlation function. And basically, it's just saying, does the presence of one of these molecules or one colored molecule in a certain spot influence the local density of the other label? So here's an example of what one of these curves might look like. And the take-home message is all you really need to know for today is that if this curve comes above 1, that tells you these two things like to be close to each other. They're enriched around one another. But if the cross correlation goes below 1, then there's some depletion, right? These things don't like to go near each other. And 1 is basically defined as just what you would expect from a random co-distribution of these two molecules. Okay, so in 2017, we published a paper where we were looking at what happens when we cluster B-cell receptors with kind of an artificial ligand. And what we found is very similar to this old story number 1, and I think it just adds kind of weight to that old idea, is that when you cluster these B-cell receptors, they stabilize kind of what looks like an ordered membrane domain. And these ordered membrane domains, they sort peptide markers of these two coexisting liquid phases. And they also sort the kinase and phosphatase that's important for this activation event. Because these phosphatases get sorted, it's sorted in a way that favors the kinase. And so this creates little hotspots for tyrosine phosphorylation. And we could see this kind of in our experiments in these B cells, but we could also recapitulate this in a kind of minimal model of this signaling pathway with kinases and phosphatases and receptors. And what's going on in this model is at time equals 0, I'm just clustering these blue receptors, and simply clustering them in the middle of the simulation box is enough to give rise to their collective activation, simply because we're changing the local concentration of these kinases and phosphatases in this heterogeneous membrane. But so after that study, we had a lot of kind of lingering questions. One was that we used this really artificial activation scheme. We stimulated with streptavidin, basically attached to an antibody that bound to the B cell receptor nowhere near its natural ligand binding site. And maybe what we saw was kind of an artifact of this very artificial stimulation scheme. So we want to know what's going to happen if we use a real ligand, a natural ligand. And also we were interested in wondering what happens if this ligand isn't soluble, if it's on a surface. Do these domains still form? Do they sort proteins that matter? And if they're there, can we tease out if they're actually contributing to the signaling process? The other kind of more technical thing that we were interested in doing by looking at this a little harder is that kind of these initial experiments in the 2017 paper were really, really hard. They were hard because the signal to noise was awful. Mostly because our counting statistics was awful, but our signal was also very small. And we were hoping that maybe by looking at this a little harder, we would also figure out ways to make this experiment easier and also more quantitative. Okay, so enter Marcos and the news story that I'm going to tell you about today. Okay, so the first thing we showed, or we realized and reproduced from the literature, is that for some reason our cells are responsive to PC lipids. And one way you can see this is by loading your cells with a fluorophore that gets brighter when it's in a higher calcium environment. And calcium mobilization is kind of a hallmark of B cell receptor activation. And so here's some movies that I'm going to show of cells landing on a surface. So now these bright spots aren't single molecules, they're actually entire B cells imaged at low magnification, and the brightness is the brightness of this fluorophore that indicates calcium concentration in the cytoplasm. Okay, so in the first experiment, we just settled these cells onto a supported lipid bilayer containing PC lipids. And you can see as soon as the cells land on this surface, they light up. And that's the indication that they're activating. I'm going to show it again because you can see these kind of cells coming into focus before they land on the surface. And that's because we've just added them to solution and they're falling just with gravity. We can do the same experiment of cells on a PE bilayer, so no PC lipids present here. And the cells settle just fine, but we don't see that kind of big spark of activation. Some of the cells do flicker, and that is actually a normal feature of these B cells. A small fraction of the population at a given time are doing what's called tonic signaling, which is a ligand-independent activation event. So we can also activate our cells that are just already sitting on a glass surface by adding a cross-linking ligand, just like we did in our first paper. And in this case, they all see the ligand at the same time because it gets washed over them at the same time. And so they have this collective kind of synchronous response. And so we can quantify this and show that, yes, the PC cells landing on the PC bilayers are activating kind of in a robust way compared to these other conditions. Another way you can show that this is a natural ligand for the B cell receptor of these cells is by labeling cells with an antibody that's specific to the activated form of the B cell receptor. So these are tyrosine phosphorylated B cell receptors. And when you label cells that landed on a PC bilayer with one of these antibodies, they light up. And so that indicates that those B cell receptors are tyrosine phosphorylated. One thing that's interesting from these pictures is that you can see this B cell, the active B cell receptor at least, is very clustered on the cell surface. And that happens even though the PC lipid is doing nothing actively to cross-link these things. It's a monovalent ligand. And so the cell is doing this clustering on its own. And how this happens is kind of well documented in the literature, but still pretty striking. So why are these PC lipids ligands? This was a question that we had. For some reason, historically, phosphocholine, which is the head group of a PC lipid, is a model ligand for B cells. So there's actually a lot of cell lines that are reactive to PC phosphocholine. But most of them are not responsive to PC lipids. For some reason, these cells are responsive to PC lipids, but not kind of the phosphocholine on their own. And this has been documented also in the literature, so it's not new at all. But it's something we can see nicely in our experiments. Okay, so now we have a natural ligand, which is great. We can settle our cells and do the same experiment we did before. And we get the same result, which is nice. So we can look at sorting of minimal peptides that mark ordered and disordered domains in vesicles. And they do sort with respect to these now tyrosine phosphorylated BCR clusters, is what we're labeling. The ordered probe is enriched in these clusters and the disordered probe is depleted, just like they would sort with respect to an ordered domain in a vesicle. And right, this is qualitatively the same as we saw before. Okay, but we ran into a big problem. And that's because these cells don't lay nicely flat on our surface. It's a technical problem. And the way you can really clearly see that is by looking at what's called an interference contrast microscopy image. And this is an interference-based method that basically reports on the distance between the top of the cover slip and the membrane that you're looking at. And so in these pictures, a darker shade means that the membrane is very close to the glass cover slip. And that happens in the same places we see the activated B cell receptors. And it's probably not surprising because those are the B cells, those are the parts of the membranes that are engaged with our surface. Right, and so we can compare our two different types of imaging and show that they co-localize very nicely, which is unfortunate to some extent because what we're doing in our normal fluorescence imaging is we're kind of assuming that the brightness of our fluorophore or the coverage sampling of our fluorophore is reporting on concentration of the thing that's being labeled. But as soon as the membrane's not flat, we lose a lot of confidence in this expectation, both because we're looking at a 2D projection of a 3D object, and that introduces its own problems. But also we're imaging in total internal reflection, which is an imaging method that is brightest very close to the cover slip. And so I just have an example of another type of cell where I think this is super obvious. So this is a fibroblast growing on a glass slide with fibronectin. And basically when you do this, these cells end up having lots of focal adhesions, which is the point of contact between the glass and the cell membrane. And so if we take a probe that we think should pretty much uniformly label the membrane, and we image it in total internal reflection, we actually see what looks like this probe being enriched in these focal adhesion sites. And you'll have to trust me that that's where they are. I don't have the other picture. We have another picture, but it didn't have room for it on the slide. But when we image by kind of off-turf excitation, so where the brightness of the excitation light isn't dependent upon how far you are away from the cover slip, the membrane label looks completely uniform. That's this high-low illumination picture. And in fact, in this example, we could image, we did some three-dimensional imaging of this probe, and you can see clearly that the points where it's brightest, kind of in the 2D image, is also where the membrane is closest to the slide in the 3D image. And so this brightness isn't an indicator of concentration, it's an indicator of location, which is not really what we care about in our other experiments. So we needed to come up with a solution to this problem. And what we did is we just made the interaction with the surface a little bit more robust by also adding in an antibody interaction to the receptor. And so that's this new example down here. And these cells are much flatter. They're much more uniform in their gray color by interference contrast. So this gives us some more confidence that with this sample prep, we can make more quantitative comparisons. So lots of caveats. One is this is a very bizarre stimulation mechanism or mode. Like this is not something you would normally think might happen in real life. So these B cell receptors can activate both because they are being clustered by this antibody, but also because they see this natural ligand. And this is a particularly artificial way of doing things. The other thing is that the antibodies and streptavidin, which we're using to make this interaction more robust, also have bulk, and those have to fit also under the membrane, and that also could be contributing to the result. I'll say so in this new activation scheme, we can still show that we're not activating receptors away from where we're labeling them by just this antibody. So it's not, I guess, so we're still getting robust activation, and the phosphorylated BCR and the BCR are all kind of co-localized, which is as expected. Okay, so we can repeat this experiment again with this new kind of activation scheme, and again, we get the same result. Now our B cell clusters, B cell receptor clusters are a little bit bigger. They take up a little bit more of the cell surface. We've added more interactions. That's maybe not surprising. They still sort these peptide markers of ordered and disordered phases. And now because these membranes are more flat, we have a little bit more confidence in the magnitudes that we get out at the end of the day. Okay, so we care about being able to quantify these magnitudes because we want to compare how these peptides sort with respect to full-length proteins. And so this is the old result that we had from before. When we clustered our B cell receptor with soluble antibodies, in particular, this phosphatase CD45 was excluded from our B cell receptor clusters to the same extent as a minimal transmembrane anchor. And this led us to conclude that that domain partitioning alone is sufficient to explain this exclusion of CD45. You didn't have to evoke some other type of interaction. We always observe that Lin kinase is more enriched than its minimal anchor, which we call PM. And this is because Lin is known to directly bind to the receptor and other proteins in this kind of receptor platform. So there's more of it around. Okay, but in this new experiment, when we activate on a surface that's both has this PC lipids and these membrane-bound antibodies, we find that the CD45 phosphatase is more excluded from these B cell receptor clusters than the transmembrane peptide on its own. And so what this means is that domain partitioning is not sufficient to describe this distribution, and so there must be other sorts of interactions that contribute to this localization. But again, Lin is still more enriched, and that's probably for the same reasons. It's still directly binding to other proteins. Okay, so this brings us back to these kind of old and good ideas. So we've kind of bolstered this old idea number one, that the ordered domains can help facilitate the kinase winning over the phosphatase by shifting local concentration. And this seems to be the dominant thing with a soluble crosslinker, and it contributes, or it also happens to some extent with a surface presented ligands, but it's not the whole story. And probably what's going on in the surface presented case is that we have something like old idea number two, where these phosphatases are occluded for some other reason. And one reason could be this kinetic segregation model, that by being close to the opposing surface, that some of these bulky phosphatases can't fit anymore, and so they also feel another repulsion. So in our experiments, we did not directly show that this was the case, we did some trying for this, but expressing these CD45 constructs in our cells was really didn't work, and so we couldn't directly test this idea. But this has been tested in other systems, in particular in the T cell system. Okay, so the real question is, so now we're saying both of these things can happen. Does one matter more than another? And so to kind of attempt to address this question, we went back to our old model. This is all a simulation now, so we can do whatever we want, right? And so in our model, when we cluster receptors, we can, the way that we cluster receptors is by basically biasing their localization to spend more time in the middle of our simulation in a circle. And so that's what's going on here. But we can do that without domains and without any other types of interactions. We can do a domain-only model, which is exactly what we did before, and where there's some partitioning based on domain preference of these components. We added in another potential. So we added a repulsion for phosphatase. So basically within the circle where we're also clustering receptors, we can say phosphatases can't go there at all. And that's what we did. And so, right, you can also activate receptors by this mechanism. And then we can combine these two effects and see what happens. Okay. So for a given receptor clustering potential, so basically some propensity for these receptors to be in this domain, we can get activation kind of in the full model where we have both types of interactions. We can get kind of a moderate extent of this activation in looking at each of these effects on its own. And we've set this up so that in a boring membrane without this phosphatase exclusion, no domains, no exclusion, these receptors don't activate just by clustering them. And to some extent, this is a choice of parameters, but it's actually pretty robust. There's a lot of parameters you could choose to get this result. And basically, the reason you get these different answers is because these different models have different local concentrations of kinases and phosphatases. This is just biochemistry. In the full model, we have no phosphatase and a fairly high kinase, an enriched kinase, and so we get the most activation. If we get rid of domains, then the kinase concentration goes down, and so does our activation. But not as many receptors get activated. And in the domain-only model, we kind of have a higher kinase, but not as low phosphatase, and so you also get an intermediate level. So nothing is particularly surprising. For me, the most interesting thing of this exercise was showing that there's kind of a sensitivity enhancement. And so basically, by combining these two effects, you can make the whole system kind of more sensitive to weaker ligands. So lower propensity to binding these receptors in the circle, just because you have these two things working in concert. So one reason you get this kind of enhancement and sensitivity is because domains help bring other receptors into this circle, even without, even when the potential is smaller. So it kind of enhances the avidity of the interaction, I think is the right word in chemistry. So I think in principle, at the end of the day, we could say combining these two mechanisms would enhance the sensitivity of cells. And experimentally, we would love to test this directly, but it's tricky to come up with a kind of experimental scheme that kind of controls all of the factors that you would like to control while you're doing this experiment. And so we didn't go that far. Okay, so along the way, we learned a few things that I thought maybe would be interesting for this crowd. One thing that kind of stood out from early on is that these bilayer engaged clusters were more effective at sorting these peptides than in our previous experiments with soluble crosslinkers. And so here the cross correlation functions next to each other, and they're actually not shockingly different, but they are meaningfully different, I think. These are for the bilayer clustered B cell receptors. these curves are further from 1, meaning that the partitioning is stronger. And one way you can see it, in these plots, you can kind of lighter colors are single cell measurements, whereas the points, bolder points are the averages over many cells. And in just looking in a single cell, you have a chance to kind of guess the answer in the bilayer presented case, but we would never think that we could even do that in the soluble case because it was just so noisy. And you can see this kind of in images themselves. So this is a nice example where the green signal is being excluded from the magenta clusters. If you squint and look hard, you can, I think, directly see this with your eyes, if you look hard enough. Right? Certainly, if you quantify it, in this particular cell, this is the cross correlation, and the error bars are small compared to the distance from 1. So that tells us it's very significantly anti-correlated, I guess. So they are not sitting next to each other. But you can't see this at all, trust me. You can't see it in a cell that's been activated by a soluble ligand. The domains are a lot smaller and everything is just harder to wrap your head around. Okay, so why is this so much easier to see? The domains themselves are larger, and this helps us in lots of ways. They're bigger than our resolution limit, which makes the intensity, helps with the intensities. They're better spatially sampled. There's more stuff in them, basically, and they're also, that just helps with our eyes. They're easier to look at. But also we find that the partition coefficient is actually higher. And even more than the cross correlation functions being a little higher, the partition coefficient, which is the thing that your eye is most attuned to, is a different number than the cross correlation function is measuring. So cross correlations measure the density kind of inside the cluster normalized by the average concentration over the whole region of interest that you're looking at. Whereas the partition coefficient is usually defined as the concentration on divided by the concentration off of the domain. And if domains make up a tiny fraction of the whole area, then these numbers are the same, and it's you get the same result. But once domains start taking up a significant fraction of the kind of membrane that you're looking at, then they aren't the same, and the partition coefficient is actually larger than the value of the cross correlation function at low radius. And so if we just plug in some numbers, kind of making some guesstimates of how much of the area our domains are taking up, so I have 1/4 here, we can turn these cross correlation amplitudes into partition coefficients. And they start being numbers that look a lot like the numbers that Ilya Leventhal's group measures for these same peptides in vesicles. So sorting is looking a lot like sorting in GPMVs, which is very exciting, I think. And so I don't know the answer of why these bilayer supported BCR clusters have this higher contrast. This is something about the physical system. Maybe what's going on is that there's just more stuff, more RAF preferring, more proteins inside these domains that kind of set how these domains, these clusters interact with membrane domains, and somehow making them stronger, more strongly kind of making it more order preferring. But we don't have any evidence for that directly. That's just kind of based on this indirect observation. So something is different about these also. They're not just bigger. Okay, but there's a downside to this observation, which is a little frustrating for us. And so the magnitude of this correlation function depends on all these details of BCR clusters. So how much of the surface they take up, probably their density, you know, these downstream things that we're not even looking at is going to affect the magnitude of this signal. And so ideally, if we'd want to do some perturbation, say change temperature or do it with some inhibitor, then ideally you need to maintain this BCR domain structure to be able to compare the results. And that's not something that's easy to do. One place this kind of showed up for us in this paper is when we ideally wanted to make the point that these bilayer supported, bilayer engaged B cell receptor clusters were more robustly activated than the soluble case. But when we do this experiment where we co-localize B cell receptor and kind of a generic phosphatyrosine stain. We got much higher amplitudes of our cross-correlation function in the case of the soluble antigen compared to the bilayer. And these are on different, have different y-axes here to hide this effect. But the reason that these are so different is not at all anything about how activated they are. It's all about the structure of the B cell receptor clusters. So in the soluble case, they're smaller. They take up less of the membrane surface, and so they give you higher. They have much higher kind of autocorrelation functions, and therefore much higher cross-correlation functions in this case. Right. So we can't just read this off and draw some kind of easy conclusion. Yes, I said that. But if you do a more subtle analysis or more kind of sophisticated analysis, you would guess that there's actually more activation in the bilayer case, and there's more co-localization with the B cell receptor clusters, and that's because the autocorrelation, the BCR autocorrelation looks a lot more like the BCR phosphotyrosine cross-correlation. And if you do FCS measurements, I think commonly when you do FCCS, so you're cross-correlating things, you always compare to the autocorrelations of the various components that you're cross-correlating. And this is why, like the cross-correlation cares about the distribution of the molecules themselves. And in this case, it's hard to kind of make up for that because there are artifacts associated with autocorrelation that we can't easily get rid of, and so we can't do such a nice analysis as is done in FCS. Anyway, it's just a technical difficulty that makes this kind of a little bit of a pain to think about how to quantify. But one more thing with this figure that I think is interesting is that so all of the time most of the time we think about, and it seems like the field thinks about, when you're clustering a receptor, you think about what's happening near the receptor. But one thing that kind of jumps out of some of our imaging measurements is that in these cells that are stimulated by bilayers, there's very little tyrosine phosphorylation happening away from the receptor clusters. They're actually, you wouldn't know a cell was there. Yeah, they just don't label at all with these tyrosine phosphorylation antibodies. But this is not the same for the soluble stimulation. There's lots of places where this phosphotyrosine antibody binds that are away from B cell receptors. And maybe this makes sense because, I mean, we'd guess since there's so much the phosphatase is being more excluded from these receptor clusters and they're taking more of the membrane, then the concentration of the phosphatase away from the B cell receptor clusters is going to be higher. And so therefore the phosphatase is going to win even more in those spots. And maybe there's some action at a distance that can matter, right? So maybe there's still proteins out there doing stuff, and maybe they're sensing this depletion of kinase and enrichment in phosphatase to somehow interact with these complicated signaling networks that govern kind of downstream signaling cellular level responses. And so I don't know, maybe there's something interesting to think about in looking at what's going on away from the receptor. Okay, just to sum up, the big kind of take-home messages is that we can activate our cells in a bunch of different ways, and we get pretty much the same result that this clustered B cell receptors seem to reside in an ordered domain, and ordered domain being defined as one that sorts peptides, phase marking peptides in ways that you would expect with respect to an ordered domain in a vesicle. When you activate with a ligand that's on a surface, the phosphatase CD45 is more excluded from the clusters that form than you would predict based on just the partitioning of a simple a membrane anchor, transmembrane anchor. And so membrane domains is probably not the whole story, and there's some other repulsive effects that are at play that have to be taken into consideration. And one candidate for that is this kinetic segregation model. And in principle, it doesn't have to be an either or. And maybe these two mechanisms actually can work in concert to make the cells more sensitive under certain circumstances of activation. And we have shown that in a model, but those experiments are a little harder to do. So kind of wanted just to demonstrate the concept. Okay, so with that, I would like to thank the people that did this. Marcos Nunez, this was his thesis, so he did the bulk of the work. At the end, we got a lot of very useful help from Kathleen Wizzer, who's a technician in the lab. And I can't underemphasize the contributions of Sarah Shelby in helping us figure out how to do these experiments and helping us understand when things don't go the way we thought. All of her contributions are completely, I don't know what we'd do without them. And then the last person I want to thank, or people I want to thank, are my kids for letting me put this talk together yesterday when I was supposed to be watching them. And also thanks to Playmobil for entertaining them for so long. Great, well, thank you.
Thank you very much, Sarah.
Yeah.
Wonderful, wonderful talk. I really enjoyed it.
Mhm.
There are a lot of questions on YouTube from the audience, but I want to start from my own question as always.
Mhm.
So how you detected CD45 is basically via the antibodies, as far as I remember from the paper.
Yes, yes.
So would it be interesting to test this kinetic segregation if this larger segregation that your PM, just to cut the extracellular part and have the TM and the intracellular part of CD45 and see still partitioning coefficient would be the same?
This would be amazing. Yes, no, so in our hands, we are not able to express CD45 in our cells, let alone manipulated versions of it. Even just the CD45 anchor, we have an enormous trouble expressing. Yeah, I would love, I mean, that is the obvious and important experiment to do. And I suspect it's not just us that has this problem. In B cells, no one's directly tested this kinetic segregation model, and usually this is the reason.
Yeah, I think
this experiment wouldn't have been done.
Yeah, in general, T cells as well as mast cells, when we tried to express CD45, it was always the most difficult molecule.
But I mean, some people have done it in T cells as successfully to really make that case strong. But yeah, we were not able to do it in our cells.
Okay, so I'll start with the audience questions. The first question is by Ilya Leventhal about this B cells and PC activating B cells. And he's asking, is this context dependent? Cholesterol or sphingomyelin blocks the binding, or maybe the proteins?
Oh.
Just wondering why these cells wouldn't be reactive to each other with PC on their membranes.
Yeah, I have no idea. I mean, so the experiment that one of the old papers that showed this is they're reactive to red blood cells. So I mean, I think they are reactive to other cells. I mean, they are a cell line, so maybe this is something that's drifted, and it's only because they're in this very non-real situation that it can even happen.
Yeah, so this probably answers the next question. Snig Hadev asking, does PC-mediated activation of B cells happen in physiological conditions? But what you're
I don't think so. Yeah.
It
sounds like a bad idea to me. Yeah, for all those good reasons. Yeah.
Okay. Another question about the KS and the domain kicking in at the same time by Ilya Leventhal again. KS and ordered domain serve same selectivity function in parallel. Why bother with both? Couldn't just make one more robust? Obviously not expecting real answer, but speculation, Ilya Leventhal.
Okay, I didn't understand fully the question. Can you say it again? Or maybe
so basically, he's asking, there's KS and ordered domain serve same selectivity.
KS, kinetic segregation.
Yeah.
Yeah.
Why bother with both? Couldn't just make one more robust? He's probably talking to nature.
Yeah, I mean, I don't know. I mean, I think one thing about immune signaling in general is that it is very versatile, right? You don't know what the ligand's going to look like, but you still want your cells to be responsive. So I think it makes a lot of sense for nature to have chosen lots of different ways to solve the problem so that it is responsive in all these circumstances. But I certainly don't know. Yeah, and there may be other things. So there's a whole class of mechanisms involving conformational changes of receptors that also, and pulling on receptors, and all of these things could also come into play. Right, under different circumstances, maybe certain ones dominate over others, but I could see why you would want it to be versatile. Yeah.
Okay. Snig Hadev asked another question. What triggers domain localization of Lin kinase upon ligand binding?
Yeah, so So we would like to think that initially, before there's kind of strong binding, before the receptors, there is very much activation of receptors, that Lin is getting there because of its domain partitioning. And that's certainly how we have it in our kind of minimal model of things. But as soon as receptor starts getting activated, Lin can actually bind to receptors and phosphorylate other receptors. And that's going to come in long before we can kind of really probe it experimentally. So the answer is I don't know, but I have a desired answer, but I don't know. We can't really test that directly.
Okay, Pablo Caravilla from Jena is asking, first saying very elegant work, congratulations. Kind of a general question, is then the hypothesis that rafts do exist, but only after cell activation? Would a resting cell have a homogeneous membrane?
So I think you could take this all in a couple of ways. So certainly that everything gets more robust once these receptors are clustered. I think that is very safe to say from lots of literature, and our contribution contributes to that. But I think you have to, I guess, define what a raft is before this cross-linking event. Like if it's a robust thing that's there for a long time, then it doesn't exist before, and that the act of clustering is actually what's stabilizing it. But if you're caring about, if you think kind of transient, changes in concentration is a raft, then I would say they're there before. And it's just that by clustering the receptor, then you're biasing those to happen more frequently at the sites of your receptor clustering. And kind of in the context of kind of phase behavior and stuff, there's kind of a way of thinking about this. Those transient things are fluctuations, and they're real when you look at a really short time scale, but not real kind of on a time average kind of thermodynamic limit kind of sense. So that's kind of where I land. I think they both exist and don't exist before, and it's all in kind of how you want to talk about what existing means.
So you basically say it's Schrodinger's raft.
Not quite, but yeah, I guess so, kind of like that. And they can form wherever you want them to. They can be stabilized in lots of different configurations and circumstances, maybe making them also very versatile.
So
yes.
Pablo's next question is also a bit philosophical. It says, if so, could two raft-mediated processes happen at the same time? Or would the cell be able to perform only one task at a time?
I think it's what's exciting. Well, one, yes, they could happen at the same time because you're not really depleting, because all of this is happening all the time, and there's kind of raft things over here and all over the membrane, and so they can be organized on their own. But I think the fun thing to think about is that this could, maybe under some circumstances you could have there be interesting crosstalk, right? By depleting the phosphatase away from the receptor cluster, then now the away from the receptor cluster knows that the receptor's been clustered, which is kind of a remarkable thing. And you can imagine that coming into all sorts of different kind of more complicated signaling networks. Kind of hurts your head to think about, but I don't know, could be exciting. Yeah.
So Adam Smith is asking, what is the distribution of receptors prior to activation? Is this distribution concentration dependent?
So as far as we can measure, and we're not as good at this as maybe others could be, but there's nothing special about the receptor distribution before activation. There's actually, we can't even measure any kind of any interesting significant correlations between receptors and our phase marking peptides before activation. But I'm sure there's lots of things in individual cells that could dictate the cellular response. The receptor concentration is one, but kinase concentration is another, kinase activity is another. Like there's all these regulatory kind of knobs that cells have to turn to, that cells could turn to tune the sensitivity, that we have no control or way to kind of measure at this point. But certainly that's happening.
Isto Kurbancic from Slovenia is asking, how is self-fixation presumably needed for super resolution imaging? Does it affect clustering, domain partitioning of proteins?
So I don't have it in these slides that I prepared, unfortunately, but we have done a lot of live cell imaging, and Sarah Shelby in particular has done an enormous amount of beautiful live cell imaging. And for everything we've looked at, I mean, we are very careful about our fixation methods, but in that context, everything that we've looked at in a live cell, we've pretty much quantitatively seen the same thing in a fixed cell. And why that's true, I don't understand. You know, each experiment has its own sets of flaws that you think could cause all sorts of problems, but they're at least different flaws. And so the fact that we see roughly, we get to the same answer in both systems, give me some confidence that our fixation isn't driving kind of the results that we're getting. But yeah, you can only do more experiments to try and bolster that idea. Yeah.
Sounds good. So Suvam is asking, if there's a possibility that lipid rafts prefers receptor clustering, is it that membrane curvature affects it too?
Most certainly could be entirely possible. Like not something we've explored at all, but there's lots of, I mean, there's some simple ways you could imagine this mattering, like say you take a protein like toxin that has some intrinsic curvature to it, but it also has a phase preference. You could imagine these two kind of interaction modes coupling in really interesting ways to drive organization. So I guess the way I think of kind of these membrane or this domain mediated kind of effects is that it's kind of one interaction mode in a long line of the different things that can happen at membranes, including curvature effects, including, you know, electrostatics, including, you know, all these known protein-protein interaction modes. And curvature is one of those too. So there could both be like, you know, separable effects, but thinking about how they could be coupled is also really exciting. And I think a new world that a lot of smart people are thinking about, but I have nothing to say about, yeah.
So have you ever tested your probes, this TR and PM probes? Do they react to curvature? If you pull the tubes, for instance, from some...
Ooh, I have not done that experiment, but if someone has, I would love to know about it. Please tell me. I mean, these are pretty boring. We try to make them as boring as possible, but that doesn't mean they're not doing cool stuff too.
Yeah. Okay. So next question is by Ilya Leventhal. I'm not going to read that question. The next question is Aurelia Honorkopf-Smith. Is there a significant difference in downstream activation between surface and soluble stimulation, like calcium levels?
So there's a lot of literature showing the surface stimulation is more robust, but what that actually means, I don't know exactly. Like, so there's different classes of downstream signaling. Calcium, I think, is pretty quantized, like releasing calcium from the ER sets the level. But there's all sorts of more subtle downstream responses, like whether the cell decides to proliferate or undergo apoptosis. That depends on all sorts of interacting signaling networks, and that I don't know if people have studied kind of in a more careful way. But it's hard, and the reviewers of this paper made this point over and over again, and I 100% agree with them that that you can't. It's always comparing apples to oranges, because you can't make the stimulus the same in these two conditions. I mean, you can use the same ligand, but it's the concentration once you put it in a membrane, like you can't do it anymore. You have to make some guesses, and anyway, so it's a difficult question to even think about how to answer. Yeah.
We have a few more questions. We're almost through, Sarah. Sorry. So Istok has another question. It's probably taken a few minutes between activation, cross-linking, and fixation. Can you speculate how early this domain is formed compared to triggering times?
Oh, Sarah Shelby would be better answering at this question than me. I mean, so in the fixed experiments, the earliest time point is about 1 minute, and that's about when calcium happens. So that's not so crazy that at least in some cells, we're not too far off, but certainly other things like downstream regulators have been recruited at this point. So the scaffolding proteins, et cetera, these are robust domains, not individually phosphorylated receptors. But I think especially in Sarah's live cell experiments, as soon as we see clustering, I think we see the sorting of these peptides. And so we can't distinguish these two events. Yeah.
Next question is by Ivan Castello Serrano. According to your results, it seems that there's a synergistic force when cluster forms. What makes raft preferred proteins be more rafty and raft excluded proteins be more excluded? Is it right?
Let's see. I think the way that we think about it is by clustering the receptor, which itself either has intrinsic kind of raft preference, or there's some interaction that it's now having that gives rise to this raft presence, but just simply clustering those without changing their preference kind of on their own is all that we need to drive this idea. Everything they're partitioning into GPMVs, we would expect them all to stay the same. What's changing is that now we've forced these receptors to all live in the same smaller area. And so that influences how likely you're to have other raft components in that area. I don't know if that makes sense. And maybe when you bring in lab in the case for B cells, and other things that also have pulmodylations that also have their own raft kind of preferences, then that kind of bolsters this environment to make it even more robust. Is kind of the way that I think about it. But the individual proteins, yeah, they don't know. I mean, they're not changing their pulmonylation state, say, because of activation. Not all of them. There are some of them. So yeah, that's actually been the thing that always bothered me about the kind of immunology raft stories models in the past, is that there was something about clustering and caused them to change their partitioning. And in our models, that's not necessary. You can have the same preference, but just by changing the localization of those same molecules, you can get rise to this effect.
Yep. Okay, next question is from Caitlin O'Brien-Bull from Simon Davis Lab in Oxford. Says, great presentation. I was wondering how you can tell whether proteins clustering affects lipid organization or vice versa.
I mean, so this is what we attempt to do. So with receptors and real signaling pathways, it's kind of hard because, I mean, when you cluster the receptor, the B cell receptor, you're also doing 1,000 other things because the cells are activating. But what we've done also is to cluster boring things, things that are like just our peptides that shouldn't have any interesting signaling effects, or just like say color toxin, which is also kind of boring. And ask the question, can just clustering those things also lead to the sorting of other phase marking peptides? And there you can see it clearly, which is nice. And you can stabilize ordered domains. You can also stabilize disordered domains. And kind of future thinking in the lab is trying to explore how general this is. Like, is this a general mode of activation of receptors that are clustered? Can clustering disordered receptors and receptors that prefer disordered environments in certain circumstances lead to their activation? There's kind of a new way of thinking about things, and lots of questions that might end up being leading in new directions or might not. But anyways, it's still a work in progress.
Okay, our last question is from Dilip from Christian Egling's lab. Excellent. Is it possible for the cell to get confused when multiple extracellular signals are present? At the end of the day, it looks like the cells need to redirect the same pool of kinase and phosphatases.
Yeah, but that's the fun. These cells are having to integrate all sorts of information to come up to make a decision, and they have lots of clever ways of doing it that we certainly haven't figured out yet. So, but yeah, I think all this crosstalk. One thing I like about this domain picture is that it does allow for some sort of interesting crosstalk between these different activation modes, and maybe that's what matters most at the end of the day. I don't know. We'll find out. Yeah.
Thank you very much, Sarah. I really enjoyed it.
Thanks for all your questions.
Audience did as well. That was amazing. Yeah. So thanks again for having time for us.
Great. Thank you.
And have a good day.
Yes, you too. See you.
Bye.
Bye.